Yes, good morning to all. In this running chapter, locomotion and movement, our next topic that is the structure of contractile proteins. If you remember those proteins which make thick filaments and thin filaments in muscles, can you recall which proteins or which protein makes up thick filaments? Myosin, exactly, Zanil and others. Good. And do you know mainly which proteins constitute the thin filaments? Actin, exactly. That's good reply. So mainly these two proteins are known as contractile proteins. Let's talk about their structure. Now here is the structure of a thin filament. This is basically structure of thin filament. In this green color structure that you are looking at is an actin filament. That one is known as which one? An actin filament. How is this formed? Have a look at it. The globular proteins, which are the monomers for the actin. Look, color has to be green. See, this is what is called as globular protein. It's a monomer. Basically, it's a monomer, single units. This one is filamentous protein or filamentous actin. So one is a globular actin, individual monomer units, and they can join with one another to turn into a polymer that is known as filamentous actin. And for this polymerization, ions which are required are Mg2 plus ions. Mg2 plus ions, magnesium ions are needed for this polymerization. Tell me, clear up to this point? The monomeric form of actin is known as globular actin. These globular actin molecules join with one another with the help of two things: energy from ATP and ions such as magnesium ion. Let them polymerize and they get converted into a filament of actin. We call it as the F-actin structure. Such, how many filaments are seen in a thin filament? Overall, what is called as thin filament. Can you make out how many F-actins are there in it? 2. Saurabh Pal, correct. This is one F actin. Can you see this thread is going? This filament is going, and entwined to that there is another F actin. So two filaments of actin entwined around each other. Entwined around each other. This is one actin filament and the other one is there, and are entwined around each other to make a thin filament. Over and above to actin filament, there is another protein which is available in a thin filament. Over and above two actin filaments, there are two tropomyosin filaments also. This one is tropomyosin, another protein. It's a sort of a fibrous protein. It seems as if there are two major F actins entwined around each other, and they are further entwined by the two filaments of tropomyosin. They run parallel to the length of actin filaments. Throughout the thin filament, you can see. That makes the thin filament. Thin filament is actually made up of two parallelly running entwined F actin filaments, and over and above to that, two parallelly running two tropomyosin filaments which themselves remain entwined around the actin filament base. Together it is called as thin filament. And another thing that you can see here, if you see carefully, a group of three small proteins, if you can make out, a group of three small proteins, that one is known as troponin. That one is known as troponin. So if I ask you a question, which proteins constitute the thin filament of muscle fiber? What shall be your answer? I asked which proteins constitute exactly actin, tropomyosin, and troponin. All the three are proteins and all the three are participants in formation of thin filament. Which proteins constitute the thin filament? Which are the different types of proteins to be found in the thin filament? Which proteins form the thin filament? Actin, tropomyosin, and troponin. These are the three different types of proteins which constitute the thin filament. Remember. Now, point number 2. In a single thin filament, how many F-actin molecules are there or F-actin chains are there? Or how many filaments of actin are there? 2. Again, in a thin filament, single thin filament, along with 2 F-actin, how many tropomyosin filaments are present? They are also 2. So 2 filaments of actin, 2 filaments of tropomyosin, and at regular interval, you have unit of unit of what is called as troponin proteins. Each one is a cluster of 3 different proteins. That's why it is called troponin, a trimeric protein. It is a trimeric protein made up of 3 different types of proteins over there. Have a look at them. Now, can you read the labeling of troponin with the enlarged version? दिख रहा है clearly? So can you make out how many subunits are there in each troponin trimer? 3 subunits. 3 subunits are there. Remember this one. You got one filament, of F-actin. This is another F-actin, you can say. Both remain entwined by what is called as two filaments of tropomyosin, and at regular interval a unit made up of 3 proteins, 3 subunits. This is called as troponin, a trimeric protein over there. In this troponin, there are 3 subunits. One of them is troponin T. One of them is called as troponin T. Another one is called as troponin I. And third is known as troponin C. Out of which troponin T subunit is a tropomyosin binding site. Troponin I is the one to inhibit ATPase activity. Now at present you will not understand these things. सिर्फ आपको information होनी चाहिए. Details अभी clarity नहीं मिलेगा आपको. So gotta wait for that. And another one is calcium binding. Calcium and will bind with this site. So this is called as calcium binding site. We can say one of them is TNC, another one may be TNT, and another one may be TNI. So 3 subunits constitute the troponin trimer, of which one is called troponin T, the one which will help in binding of tropomyosin, which will help in binding with tropomyosin. Another one, TNI, the one which can inhibit the ATPase action. And third is troponin C, TNC, the one which can receive calcium ion. It has a calcium ion binding site on it. Tell me, is this clear on your part? This makes up the whole thin filament. A rapid fire round. A thin filament which is found in A band and even I band arises from the middle of I band, that is Z line, passing over the entire remaining half of the I band. It further penetrates into A band, and at the periphery of A band, it will overlap the thick filaments. These thin filaments are made up of three different types of proteins. What are the names of these three different types of proteins which constitute the thin filaments? Actin, tropomyosin, and troponin. How many filaments of actin will participate in making of thin filament? Two, correct. Then which is another filament which are also two in number and are participating in formation of thin filament? Tropomyosin, isn't it? The other one is a tropomyosin. And last, which is the trimeric protein that is found in the structure of thin filament? Yeah, it's a globular trimeric protein occurring all along the length of thin filament at regular interval. And it has got three subunits. What's name of first subunit? TNT, troponin T, that is the site where troponin will attach with tropomyosin. Which is the another one? TN1, I rather, what one? TNI is standing for the one which will inhibit the ATPase action, which you learn later on. And which is the third one? TNC. Which ions are going to bind to it? Exactly, calcium and Rudra and others, correct. So troponin C stands for the site of the troponin or subunit of troponin on which calcium binding sites are there. So that is termed as TNC. अच्छा, ये जो ट्रोपोनिन होता है, जब इसके ऊपर कैल्शियम लगता है, when calcium ions bind to it, at that time it will make the tropomyosin to slide a little bit or to move away a little bit and expose specific sites that are present on F-actin, the sites which are known as myosin binding sites, the one where myosin can attach to the actin. That site on actin is called as myosin binding site over there. Okay, these are the active sites where myosin can bind on the actin filament. We would say that जैसे ये actin है, इसके ऊपर क्या लगा हुआ है? Tropomyosin. It is present on it. Look at the second step. When calcium binds to this, to troponin, TNC, that will lift these tropomyosin a little bit and exposing specific spots, unmasking, mask and unmask. आप अपने मास्क कवर करते हो तो योर फेस इज कवर्ड, मास्क उतारते हो तो योर फेस इज अनमास्क्ड, ओपन अप. जब तक ट्रोपोमासिन था on the affecting, the myosin binding site present over here. They are called as myosin binding site on F-actin molecule. These sites were masked, but the moment the myosin is lifted, these sites are open. Now this F-actin can attach with myosin. Now the thin filament and thick filament can attach to each other and muscle contraction can start.
Sir, your
As
voice
soon
is
as calcium.
cracking.
Yes, sir.
Yes, sir.
Okay, hold on. Or just give me a minute. Hello, am I audible?
Yes, sir.
Yes, sir.
Now, all of you pay attention. I'm repeating. This is the most crucial point in this contractile proteins functioning and structure and function का जो एक important aspect है वो मैं आपको explain कर रहा हूँ. Please pay attention and understand. I want all 50 of you to understand this line. Before the calcium ion came into picture, the F-actin filament, it was covered with tropomyosin, but the moment calcium ion binds with troponin, particularly TNC subunit of it, its position changes a little bit, so that attached tropomyosin is lifted a little bit up. When it is lifted, immediately certain sites which were present on actin filament earlier were masked but now are exposed. The moment they are exposed, these sites are known as myosin binding sites. The sites where myosin can bind over there. इसके साथ myosin filament bind कर सकता है. These sites are known as myosin binding sites. They are actually present on actin filament. Earlier they were masked, M-A-S-K-E-D, masked. Because of calcium availability, troponin came into action, lifted the tropomyosin, exposed or unmasked the myosin binding sites on actin filament, and now the myosin can attach with them and can start the process of muscle contraction. How many have understood? 1, 2, 3, Gallows, Anil, Jugal, 3 only. Aksa and Jayati and Saurabh and Aneri and Mutahir and Khushali and Afia, Yug, Atre, Mahavir, Sri, Shreya, Kashish, Ausar, Adrian. Fine, many could understand. Now I want the ones who have not understood to raise the hand or to write down not clear. Okay, nobody did this thing. That means majority have understood. मैं आप लोगों को पूछता हूं, आप लोग आंसर दो. Step 1, calcium comes and binds dash. क्या लिखूं मैं? TNC part of troponin. Excellent. So that the conformational change takes place in troponin and it lifts dash. Exactly, it lifts tropomyosin. This exposes dash sites The unmasking of dash sites takes place because of the myosin binding sites, which are actually present on dash. They were present on actin filament. Zenil was the one. Wow. So now what can bind to these actin filaments? What can bind to these actin filaments now? Exactly, Galav and Zenil. Myosin can bind to this one. And that will allow the beginning of which process? That will allow what to start? Exactly, Galav and Zenil again. Wow. Muscle contraction. It seems majority of you have understood. Before calcium ions came in, which protein had covered up the myosin binding sites of F-actin? Troponin and tropomyosin. They were ultimately responsible for that. The moment calcium ion came in, binded with TNC of troponin, lifted the tropomyosin from its position, exposing the myosin binding sites present on F-actin, allowed myosin to bind over there with F-actin. Thick and thin filaments attach हो जाते हैं and process start हो जाता है. Name of that process is muscle contraction. Which are the other filaments other than thin filaments of myofibril? Which are the other type of filaments? Exactly, thick filaments. And only one type of proteins constitute the thick filaments. Unlike that of thin made up of actin, tropomyosin, troponin, the thick filaments over here are made up of only one type. Which is that protein which constitutes them? Exactly, that's the myosin. Only one, myosin. Myosin protein. Myosin filaments make it up, make this thick filaments. If you see the myosin, now let us go in detail of the structure of thick filament. Very important and complicated. Please understand the same. First of all, what happens is that meromyosin is a monomer which polymerizes to what is called as myosin filament. यहाँ पे दो meromyosin दिखते हैं आपको snake जैसे. Are you able to see that?
Yes, sir.
Two meromyosin units as a monomer entwined around each other as if there are two snakes over there. ऐसा लगेगा आपको apparently. Okay. Now what you are looking at is myosin filament. These were what? Meromyosins. Individually, these were what? Meromyosin. But as a whole structure, if you see, then it is a polymer of so many meromyosins that is called as myosin filament. I hope you all have understood. What you are looking at is not a myosin, you are looking at the just a pair of meromyosin. दो meromyosin को आप देख रहे हैं. ऐसे बहुत सारे meromyosins मिलके क्या प्रिपेयर करते हैं? Myosin filament. I'll show you exactly how should it appear. Look at this one. These two, if I point out, they are what? They are maromyosin, two only. But this stuff is known as what? This is, I should be labeling as what? Tell me. The whole stuff is called as what? Exactly. You guys are intelligent. It's a myosin filament. ये एक monomer था, maromyosin. Whereas myosin filament, myosin is a polymer. Always remember this. Maromyosins, this is the myosin. देखो बहुत सारे maromyosins मिलके बना रहे हैं myosin का filament. That is called as a thick filament. This is called as a thick filament, also known as myosin filament. I hope you must have understood the same. Now I'm going to explain to you the individual maromyosin structure. देखो ये जो एक सिंगल स्नेक जैसा है ना, that is a maromyosin. Generally they are found in a pair. They are found in a pair. If you see, there are two parts like A part and B part. A part is within this maromyosin. A part is the heavy maromyosin component. इसको HMM कहते हैं. A part को क्या कहते हैं? HMM कहते हैं. HMM stands for heavy maromyosin. This much is HMM. अच्छा, ये जो होता है सेकेंड पार्ट, उसको कहते हैं LMM. LMM stands for what? Light maromyosin. Light maromyosin. ऐसा. इसको HMM कहेंगे, इसको LMM कहेंगे. अच्छा, इसमें इतना पोर्शन है, उसको हेड कहते हैं. बाकी का जो पोर्शन है उसको क्रॉस आर्म शॉर्ट आर्म कहेंगे. क्रॉस आर्म नहीं, शॉर्ट आर्म कहेंगे. और इसको नॉर्मली टेल कहते हैं. गॉट इट? और ये हेड और शॉर्ट आर्म को मिलके क्रॉस आर्म भी कह सकते हैं. इफ यू सी द टेल्स आर एंटवाइंड विथ ईच अदर, you can make out the tails are entwined with each other. Now, first of all, I should explain to you the structure by confirming whether you have understood or not about this structure. This is one maromyosin. Now, if I ask you this much portion, what is the name of this portion? Short form, LMM or tail? This is called LMM or it is called tail. Okay. Light maromyosin. What do you call this remaining stuff as? HMM, heavy maromyosin, या कहते हैं क्रॉस आर्म. This is called as cross arm. Okay. In this cross arm, this portion is known as what? Head. Too good. You guys are doing well. Too good. Head. This much is known as what? Nice. That's a good reply. That's called short arm. So the whole molecule is called as meromyosin. This meromyosin has two components, HMM component, which is known as a cross arm, and LMM component is known as tail part. Cross arm and tail, या फिर HMM and LMM. Heavy meromyosin, light meromyosin. फिर ये जो HMM है उसके भी दो सब पार्ट्स, head region and short arm. Together both are even known as cross arm. So now I think the structure is clear, so I can take you back to the further details of that meromyosin unit. In the head region, as you can see, there are two important sites. Which is that site? Have a look. Actin binding site. That site is termed as which one? Actin binding site. Acha, actin binding site is present on myosin filament. And these sites exposed on, what are these sites known as? Tell me. ये जो unmasked sites दिख रही है आपको? Exactly. Myosin binding sites, these are. ये किसके ऊपर होती है? एक्टिन फिलामेंट के ऊपर. एक्टिन बाइंडिंग साइट्स किसके ऊपर होती है? मायोसिन फिलामेंट के ऊपर. Interesting, na? Actin possesses site where myosin can bind. We call it as a myosin binding site. Myosin filament possesses site where actin can bind. We call it as the actin binding site. और जब मसल कॉन्ट्रैक्शन होता है तो ये दोनों का कनेक्टिविटी हो जाता है, ये लिफ्ट हो जाएगा पूरा का पूरा आपका क्रॉस आर्म और वहां पे अटैच हो जाएगा. एंड देन मसल कॉन्ट्रैक्शन शैल बिगिन. होली क्लियर यहां तक? हैव यू ऑल अंडरस्टूड? एक्टिन बाइंडिंग साइट का कंसेप्ट क्लियर हुआ आपका? अच्छा, additionally there is one more site over there, and that site in the head region is called as ATP binding site over there. It is known as which one? ATP binding site. Muscle contraction requires a lot of energy provided by ATP, and these ATPs bind over here and undergo breakdown into ADP and phosphate. This site acts like an enzyme. It is ATP is enzyme. ये साइट एटीपी का ब्रेकडाउन कर सकती है. इस साइट को एटीपी इज एंजाइम साइट भी कहते हैं. सिंप्लीफाइड टर्म्स में उसको एटीपी बाइंडिंग साइट कहते हैं. इसीलिए मायोसिन एक स्ट्रक्चरल प्रोटीन है जो मसल्स के स्ट्रक्चर में इन्वॉल्व होता है, एज वेल एज इट इज अ फंक्शनल प्रोटीन जो एक एंजाइम एक्टिविटी को भी परफॉर्म करता है. दिस वन इज नोन एज myosin ATP binding site. Now, as I mentioned earlier, that many such meromyosins polymerize themselves to make a thick filament. In this thick filament, as you can see, I'll show you in the pic. Can you make out? so many HMM of various maromyosins. Can you make out? ये सब क्या है? HMM parts are projected out from the thick filament. Thick filament mainly tail से बना और HMM parts are projected outward and at angle from the myosin filament. एक angle create करते हैं myosin filament में. वो filament simply run कर रहा है like this. और उसमें से angle जैसे बाहर निकलते हैं. So at angle, they maintain an angle while they are projected out from the surface of the myosin filament. And this is done at regular interval. These are called as cross arms. Yes, these projected units are called as cross arms. Very important topic, cross arms. Cross arms. In a thick filament made up of myosin, from the myosin filament itself, in pairs, the cross arms are projected out at angle to the filament at regular distance. These are called as cross arms. HMM components of maromyosin project out at angle from the main thick filament. These projected structures are called as cross arms. Another name of HMM is cross arms. Do you understand? With this, I conclude the topic of structure of contractile proteins. That topic is over. We shall have a break of few minutes and then we resume our work. Contraction of muscle. That's our next topic. So for next 5 minutes, enjoy the break. So to understand the process of muscle contraction, first of all, you should know that there is one theory which we got to understand that will explain how the muscle contraction occurs. That theory is called as sliding filament theory. This theory actually explains how muscle contraction occurs, how muscle contraction takes place, how muscles contract. That can be explained by one theory, which is called as sliding filament theory. It explains the mechanism of muscle contraction. Muscle contraction mechanism can be explained by sliding filament theory. This theory basically, it explains that while muscles contract, while muscles undergo contraction, actually they do not contract themselves. They slide over each other and make the muscle fiber to undergo contraction. As and when it will come, the later part, I will explain what it is. आप सिर्फ इतना याद रखो at this point. The theory by which you can understand how muscles undergo contraction, that mechanism you can understand, is called as sliding filament theory. It believes that the thick and thin filaments do not contract, they do not reduce their own length to reduce the muscle fiber length and contract it. They slide over each other and then the length of the muscle fiber decreases. जैसे कोई किसी यहाँ पे अगर ये thick filaments है और यहाँ पे वो thin filaments है, so ऐसा नहीं होता कि thick and thin filaments अपनी जगह पे contract हो जाए और length short हो जाए. No, it doesn't happen like this. It says that thin filament or thick filament में thin filaments will slide within the thick filaments. Thin filaments will slide within the thick filaments and will reduce the length of the muscle. I'll explain as and when it will come later on. For time being, just itna yaad rakhna, sliding filament theory we are trying to understand. We'll go back to the diagram which we had earlier. We must first of all understand the connection between the nervous system and muscles, because nervous system only can instruct muscles to undergo contraction. So here you can have a look that there are several muscle fibers are there, on which you can see your nervous system is connected. Nervous system has a connection with these muscle fibers. We say innervation of nervous system in the muscles. The term used is innervation of the muscles with the help of nervous system. This is generally मोटर न्यूरॉन, ये जनरली नर्व एंडिंग है, तो ये होता है एक्सॉन ऑफ मोटर न्यूरॉन. First of all, nerve cells are neurons. There are two types of nerve cells, sensory neurons and motor neurons. Sensory neurons convey the information from the structures of the body back to the CNS or nervous system, whereas motor neurons pass on the messages from the nervous system to the body structures like muscles and other things. So ये messages को muscles की ओर लाएगा from the central nervous system, तो इसको मोटर न्यूरॉन कहते हैं. और जब कोई भी न्यूरॉन होता है उसमें तीन पार्ट्स होते हैं. एक होता है उसके डेंड्राइट्स, एक होता है उसका साइटॉन, और बाकी का लॉन्ग एक्सॉन होता है. An axon is the one which is gonna terminate on muscles and glands and any other such structures. So basically you got to remember that it is the axon end of the motor neuron terminates or innervates into the muscle fiber. ऐसा याद रखने का है. अच्छा, where it will terminate, it will end up. This is the axon end, axon of the motor neuron. This is its end part. This end part of the axon, it is known as synaptic knob. It is known as what? Synaptic knob. Synaptic knob कहते हैं इस पूरे यूनिट को. That is called as synaptic knob. Remember this one. So the motor neuron's axon terminates into a muscle fiber by its synaptic knob. The part where it is coming in contact, it is supposed to be the layer, the cell wall of the cell membrane of the muscle fiber. What's the name of the cell membrane of the muscle fiber where this synaptic knob will communicate? That is termed as sarcolemma, where it will terminate. This synaptic knob, the end of the axon, like this. And can you see the small receptors on the surface of sarcolemma? Can you see that? What's the exact location of these receptors to over there? They are present on the surface of sarcolemma. And can you make out that in the synaptic knob There are small vesicles filled with chemicals called as neurotransmitters. Example, acetylcholine करके एक acetylcholine, that's the name of neurotransmitter. So neurotransmitters such as acetylcholine over there. Can you make out these vesicles over there? So यहाँ से वेसिकल में से, जैसे आप इमेजिन करो यहाँ पे मैसेज आता है, फिर यहाँ से रिलीज होता है न्यूरोकेमिकल, और वो जाके किसको बाइंड करेगा? रिसेप्टर्स प्रेजेंट ऑन सार्कोलेमा, और मैसेज यहाँ पे पहुंच जाएगा. Impulse will reach to the muscle, because between the axon and the synaptic knob and the surface of muscle fiber, that is sarcolemma, can you see there is a physical gap between the two? Can you point out that gap? This gap I am talking about, a physical gap between synaptic knob and sarcolemma, जिसको synapse कहते हैं. उसको क्या कहते हैं? साइनैप्स कहते हैं. उस गैप को साइनैप्टिक गैप या साइनैप्स कहते हैं. Always remember this one. So where there is a physical gap, how do you convey the message or impulse from one that is axon and to the sarcolemma? So that is done via neurotransmitters. Between these gaps, neurotransmitters are released. This setup is known as motor end plate, या फिर कहते हैं उसको न्यूरोमस्कुलर junction. This is termed as which junction? Neuromuscular junction. How many are clear with neuromuscular junction? The motor neuron ends up into the, or innervates into the muscle through its axon terminal known as synaptic knob on the sarcolemma of the muscle fiber, maintaining some space called synapse. This connection is known as neuromuscular junction, या फिर motor end plate. It is known as motor end plate. ये एक न्यूरोमस्कुलर जंक्शन है, ये दूसरा न्यूरोमस्कुलर जंक्शन है. जहां पे भी एक्सॉन टर्मिनेट्स इनटू द मसल फाइबर विथ इट्स सिनेप्टिक नॉब, coming in contact with the sarcolemma by maintaining some physical space over there called synapse, the communication junction of this category is known as neuromuscular junction. So when message comes at the synaptic knob, which vesicles release a chemical? These vesicles are called as which vesicles? Synaptic vesicles. And the chemical present in them are known as which chemicals? Synaptic knob में जो vesicles होती है, they are called as synaptic vesicles. And what chemicals present are known as neurotransmitter. Yes, Daniel, correct. And which neurotransmitter is normally present at the neuromuscular junction? Exactly, acetylcholine. And the receptors for acetylcholine are located at which position? रिलीज होने के बाद ये न्यूरोट्रांसमीटर जाके किसको बाइंड करेगा? उसके रिसेप्टर को. सो वो ACH, acetylcholine receptors are present on sarcolemma. Exactly. So now I'm going to explain to you about the process, the beginning of muscle contraction, the first half of it. So from where the impulse will be released to stimulate muscle contraction. An impulse to stimulate muscle contraction shall be released from where? Exactly, CNS. That's a step one. Which kind of neuron carries this impulse from the CNS to muscle? Fabulous motor neuron. An impulse generated for muscle contraction by CNS. It is relayed by motor neuron. Which part of motor neuron terminates into motor end plate? Axon. Specifically, which portion of axon? That swollen button like or bulb like, which part of the synaptic knob? Excellent. This impulse will stimulate what? Release of neurotransmitter from synaptic vesicles. Jordar. This will lead to release of which neurotransmitter at the synapse from these vesicles? Exactly, Jugal. Acetylcholine release takes place over there. This acetylcholine is gonna bind to dash. Acetylcholine receptors. Very nice reply. Where are these receptors present? Exactly, on a sarcolemma. This generates action potential. Which will spread through the sarcolemma into the muscle fiber. Action potential का meaning पता है? Simple meaning तो है stimulus. So एक stimulus generate होगा जो spread होगा वहां पे. बोलो ये steps कौन revise करा देगा? Step number 1, जुगल गालाव, who will try? गर्ल्स में से कोई हो तो कैपेबल? बहुत सारे हैं क्या? गालाव, जुगल, श्रेया, good, 3 are there at least. गालाव, टाइम नहीं है, शॉर्ट में करना है, फास्ट करना है. First,
Yes, sir. From CNS, the message for muscle contraction is released, which will be taken by the motor neurons. When the motor neurons reaches to the muscle fibers, their axon
नहीं, motor
and
neuron reaches to the muscle fiber नहीं. You have to say impulse via motor neuron will reach to the muscle fiber.
okay, sir. The impulse via motor neuron when reaches to the muscle fiber, the axon end of the motor neurons will be converted into synaptic knobs.
No. It will not be converted. It is already there as it is. It is the synaptic knob which will have vesicles and that will be releasing
Neurotransmitters.
named
These neurotransmitters are released. Neurotransmitter include acetylcholine, which will bind with the receptors which are present on sarcolemma of the muscle fiber, and now these neurotransmitters will generate an action potential due to which the muscle contraction will take place.
Have you all understood?
Yes.
I revise and then I go to the second sub part in the first half of the muscle contraction. Step number 1, CNS releases an impulse for muscle contraction. Second, this impulse is conducted via motor neuron till the neuromuscular junction. Third, the axon end, which is called synaptic knob, on receiving the stimulus releases neurotransmitter from its synaptic vesicles. These neurotransmitter acetylcholine via synapse reaches to the receptors, acetylcholine receptors, to be found on the surface of sarcolemma at neuromuscular junction. This generates an action potential which will spread in the muscle fiber. आगे का story further from this point. You have already learned up to this point, isn't it? Now, from this sarcolemma, this is the sarcolemma. Can you make out this sarcolemma? This sarcolemma. So can you make out the sarcolemma? In which can you make out the inverted projection over there? What should this one be known as? ये जो छोटा सा प्रोजेक्शन होता है जो सार्कोलेमा में से सार्कोप्लाज्म की ओर होता है, इस प्रोजेक्शन को क्या कहते हैं? T tubules. जुगल. Very good reply. ये तो था आपका न्यूरोमस्कुलर जंक्शन. फाइन. Synaptic knob with neurotransmitters and sarcolemma. But from these sarcolemma, you can see the inner structures called as T tubules. It is in contact with the structure that can release calcium, the calcium storage site. What is what is the structure known as, which is storing the calcium and gonna release the calcium soon? बोलिए ना. जुगल. गलत. टीएनसी तो वो
सार्कोप्लाज्मिक रेटिकुलम.
एग्जैक्टली. एसआर मैंने एक्सप्लेन किया था कि इसका जो सार्कोप्लाज्मिक रेटिकुलम है ना, वो इंपॉर्टेंट है, वो कैल्शियम को स्टोर कर रखा है. सो दैट इज द वन. ये तो था टी ट्यूब्यूल आपका, और ये है सार्कोप्लाज्मिक रेटिकुलम. सो मैसेज व्हेन रीचेस टू द motor end plate, neurotransmitter release goes and binds to the sarcolemma where the receptors are there. This will generate an action potential via T tubule. This message goes to the, this stimulation goes to the SR, and in response SR shall liberate calcium ion. SR will, sarcoplasmic reticulum will liberate calcium ion. These calcium ions will go and bind within the muscle fiber. They will go and bind to which specific protein? And it is to be found in thick filaments or thin filament. इसमें जाके bind करेगा वो? TNC subunit of troponin on thin filament. Galav, Shreya, both are collectively correct. This calcium will go and bind to TNC subunit of troponins which are scattered on the thin filament. Thin filament के troponin trimers में से TNC को जाके bind करेगा. That will unmask the myosin binding sites on actin filaments, and this will allow the cross arm of a thick filament to bind with myosin binding sites of thin filament. ये तो याद था ना आपको? कैल्शियम रिलीज होने के बाद क्या होता है? You remember? अब वो पीस ऑफ इंफॉर्मेशन को यहाँ पे इंसर्ट कर दो, तो एक कंटिन्यूअस फ्लो क्रिएट होगा आपके माइंड में। ऐसे होगा, CNS releases an impulse for contraction of muscle fiber, which is conducted via motor neuron, bringing that impulse to the synaptic knob, the axon end. The synaptic vesicles in turn will release acetylcholine, a neurotransmitter inside the synapse. This goes and binds to the receptors present on the sarcolemma. This binding of acetylcholine to the receptors on sarcolemma generates an excitation, or you may call it as an action potential. Via T tubule, this action potential reaches to the sarcoplasmic reticulum. In turn, sarcoplasmic reticulum will release calcium ion, which were stored in it. This calcium ion will go into TSC cell of Troponin primers on the thin filament. Removal or shifting of tropomyosin and opening up or exposing the, you can say, myosin binding sites on actin filaments, and immediately these cross arms of thick filaments will bind with the thin filaments, and जैसे इन दोनों का interlocking होता है, interlocking between cross arm of thick filament and myosin binding sites of thin filaments, now the muscle is beginning to contract. Muscle contraction से पहले का जो एक बैकग्राउंड क्रिएट करना होता है, वो मैंने एक्सप्लेन किया। आपको पता चला क्या? Did you understand the whole thing or not? Okay, now hardly 15 minutes are left, in which the remaining contraction process I cannot explain. I cannot conclude it on time. So I would say, let's have a revision. Ma'am will revise in 15 minutes, 5 minutes for actin filaments, 5 minutes for myosin filaments, and 5 minutes for this sliding filament theory. First half up to this cross bridge formation. ये जो cross arm attach हो जाता है ना actin को, then after it is no longer called cross arm, it is known as cross bridge. Cross bridge formation तक का मैडम आपको पढ़ाएंगे. Fine, मैडम.